Um, Good afternoon, everyone. Welcome to this week's Endocrinology Grand Rounds. And we are very pleased to have Dr. Deng talk to us today. Dr. Deng is currently Associate Professor in the Department of Surgery in the Division of Immunotherapy at the Brown Cancer Center and University of Louisville. Dr. Deng obtained his initial training in China, where he did his Bachelor of Science degree, his MS, and also his Ph.D. He came to the United States to Georgia, where he was at the Center for Biotechnology and Genomic Medicine. He did a postdoc fellowship at University of Alabama at Birmingham before coming to the University of Louisville. He came here as an assistant professor on a tenure track, has been very successful, has been a tenured associate professor since this year. And he currently has 3 R01s as a principal investigator, and he's a co-investigator in 3 additional R01s. He has extensive publications record and is highly respected for his research work. And his research focuses on understanding the regulation of gut exosomes, inflammation, and gut microbiota, and colitis, and their link to development of cancer, particularly colon cancer. And today he's going to talk to us on a related topic, which is the gut-liver axis in steatotic liver disease, something that has been of growing importance, both metabolic and alcohol-related steatotic liver disease are now very prominent in terms of their rapid advances in diagnosis and treatment. And I think his work will be very important in the future for us to develop new approaches to their management. So thank you, Dr. Deng.
Thank you, introduction. So today I'm very happy to present my work. My research is focused on the sphingolipid and the immune regulation in the alcohol-related liver disease and non-alcohol-related liver disease. They just changed the name, the MAS-ALD and the MET-ALD. My research also focused on the colitis and the colitis-associated colon cancer. So in the gut, there's a epithelial is a very important several type of the IgC can secretion the high secretion activity, like the Panzer cell can secrete the antibacterial peptide and neutralize the antibacterial, and like the goblet cell also can secretion the mucus. A mucin can form the mucus, can separate the commensal bacteria and from the veni and also pathogenic bacteria. And in the gut, there are very important immune system, including the B cell, can secretion soluble Ig, can neutralize the commensal bacteria or pathogenic bacteria. So there are also the macrophage and the DC in the gut. They can sample the food antigen and the nominal bacterial antigen and process this antigen to present this antigen to the T cell. Sometimes can activation T cell and keep the immune system homeostasis. But in the high fat condition or the alcohol feeding the condition, like the high fat diet and high fructose and ethanol diet, usually that can cause the intestine the dysbiosis, cause the bacterial overgrowth here, and most can reduce the mucus and promote the gut leaking. And this the leaking gut will promote the bacterial translocation to the liver and cause the liver injury, because there's any gut-derived factor can activation the liver cell, the immune cell is a liver environment. So the alcohol diet and the high fat diet usually causes very lower grade inflammation in the gut. So we take a look at the intestine. We look at the IgC. On the top of the IgC, there is a mucus. This mucus is secretion by the goblet cell, promotes the mucus formation. There is a two layer. One layer is the outer mucus layer. Another layer is the inner mucus layer. The most of the plant cell secretion antibacterial peptide, like the defensin, like the recognin-3 beta, they can antibacterial in the outer layer, but the IgA, soluble IgA, usually here in the inner mucosa. So usually the bacteria, or commercial bacteria, or the pathogenic bacteria cannot pass through the inner mucus. And some bacteria pass through the IgC. We call the specialized IgC, called the MCL. They can process this bacteria and uptake by the dendritic cell. This DC can process the bacterial antigen, then migration to the mesenteric lymph node. Here they can activation T cell in the live T cell in the mesenteric lymph node. Once T cell activation, they will migration back to the gut, and then activation helps the B cell secretion soluble IgA. So this IgA is a T cell dependent IgA. So this IgA can has a high affinity to neutralize the pathogenic bacteria. When you use the bacteria probe to detect the bacteria, we use a 16S bacteria to scanning the gut lumen. You can clearly see the bacteria can separate from the veneer. There is a space about the 50 nanometer mucus. So usually in the live condition, the bacteria cannot attach to the epithelial cell. So in the gut lumen, the goblet cell not only secretions the mucus, we also can find some small particle in the gut lumen. Like here, you can see these goblet cell. On the top of the goblet cell, you can see many small particles. We call the extracellular vesicle because the has some 20 nanometer and 200 nanometer. Very looks like the exosomal like the particle. And further, we can find this particle usually attached to the bacteria and close to the veneer. This data suggests maybe the gut-derived particle can interact with bacteria and help maintain the gut homeostasis. Again, when you use antibody, we call the CD63 is antibody exosomal marker can scanning exosomal, or we call the extracellular vesicle. When scanning, you can find this 16S RNA is a proper that detects the bacteria. You can find these two particles can interaction with the bacteria in the normal mice, their space. But under the high fat condition, this space was destroyed. Many bacteria attach the veneer, and also the extracellular vesicle can interaction with bacteria. So under the high fat condition, or the alcohol condition, we think because the gut and the liver crosstalk every day, the liver can receive the signal from the gut, like the ARPS or food antigen or bacterial antigen. We propose that liver also can maybe receive the signal like the GV carry the signal communication with the liver. So maybe under the alcohol condition or under the high fat conditions, they carry the different signal in the GV. So to find such a GV function, we label we collect the extracellular vesicle and label the dye and with the mice. You can find this particle can go to the liver. Usually they can pass through the lymphatic capillary. You can hear is a red color. It's also can pass through the blood vessel. The CD31 is the blood vessel and the UV standing by the green. You can clearly see the particle pass through the vessel. When this particle go to the liver, they can uptake by the macrophage. The F40 is the macrophage marker, is we call also maybe is a Kupffer cell in the liver. So this data suggests the gut-derived UV can communication with the liver. So to find the function, we use one model is a Conway-induced liver injury model. When you inject Conway, they can damage the liver, induce the liver hepatocyte damage within 24 hours. But usually this Conway is mediated by the NK and NKT cell because the Conway can activation NKT and NK cell. Once NK or NKT activation, they can secretion lots of cytokine intergamma. Gamma. Their intergamma can damage the hepatocytes. Usually, NKT the percentage in the liver in the immune cell about 20 and 30%. But when you injection Conway, they can cause the NKT activation and they die very quickly in the 8 hours and cause the liver injury. And in the 24 hours, the NKT still lower than is 1%. However, the 3 days later, they can recover. But pre-treated with this particle that can prevent the liver injury. Here you can see the NKT lot of activation. So we call the naive, the gut-derived naive, the UV can induce NKT tolerance. That means because the liver every day receive the signal from gut, maybe should induce the immune cell tolerance. Otherwise, they will cause the inflammation. So here is we propose the gut-derived particle communication with the liver and uptake by the Kupffer cell or DC. They present this signal to the NKT, induce NKT activation. So because the UV is complex, they contain high lipid and protein and nucleic acid. So we want to which component induce such NKT tolerance. So we do the lipid profile, omics, and find the many lipid contains UV. We call the PG2 or S1P. S1P is a sphingolipid. So our research is focused on sphingolipid in the alcohol and non-alcohol liver disease. So what's the sphingolipid? And ceramide is one of the very important sphingolipids that can derive from the dietary sphingomyelin and degradation by the SMAS, this enzyme. And the ceramide also can degradation by the ceramides to produce the sphingosine and the fatty acid. These ceramides include the 3 ones, acid and neutral and alkaline. And the sphingosine also can phosphorylation by the sphingosine kinase, produce S1P like the SK1, SK2. Once S1P ligation to the S1PR1, and there's a receptor, there are 5 receptor. Particularly S1P ligation S1PR1 that can induce Th17 migration. We do some work, the S1P and S1PR1 signal is the Th17 in the colon cancer. Our work is focused on here is the S1P in the alcohol liver disease, in the liver disease SK1, SK2. We also interested into the ceramides, like the nuclear ceramide in the obesity and the NASH. And we also do some work in the sphingosine in the gut infection. So we think the high fat diet or the alcohol diet can induce intestine inflammation. And this lower grade intestine inflammation maybe contribute to alcohol liver disease. We feed the mice with alcohol diet that can cause the very lower grade intestine inflammation. You can see the large intestine and small intestine, many immune cell infiltration to the IgC. When you analyze this immune cell, we analyze the lamella propria lymphocytes. You can see the Th1 is accumulation in the large intestine and the small intestine. You can see here compared to the Ph.D control dye, the Th17 is increased. And also find the our gamma T, this population also increased. Because our gamma T is a transcription factor contribute to the Th17 development, you also see the population is accumulation. This data suggests maybe the Th17 and our gamma T CD4 T cell accumulation in the gut may link to the intestine inflammation. So, and then contribute to the LD development. So we wonder Th17 is contributed to the LD development. We use our gamma T knockout mice. So because our gamma T is a master regulator of the Th17 cell, this mice and knockout this gene, so to prevent the Th17 in the development. So compare the white mice after alcohol feeding. Our gamma T development is very less, liver injury, or standing, and the Qi. When you look at the intestine, there our gamma also very low, the intestine inflammation. This data suggests the Th17 can contribute to the LD development. So the question is, how the Th17 accumulation in the lamella propria in the LD development. So our several years of our work find the S1P ligation to the S1PR1. They can cause the Th17 migration because S1P is a very important inflammation lipid. They can cause the immune cell migration out from the lymph node to the blood or other organ. So we guess maybe S1 sphingolipid is very important in the LD development. So next, we profile the sphingolipid metabolites in the LD patient with moderate and severe patient. So you can find the serum ceramide is a huge increase compared to the health control, and serum sphingomyelin also increase. We also check the two enzymes, SK1 and SK2. These two genes control the S1P production, and is also high expression in the large intestine and the small intestine. When you do the profile, gut ceramide, you also find the ceramide is a high increase in the gut and including the sphingomyelin. All of the data suggest that the sphingolipid, including the S1P, may be contributed to the LD development. So we use the SK1 knockout mice, feeding the alcohol dye. Compared to white mice, this deletion SK1 can significantly decrease LD alcohol-induced liver fatty by the reduced red oil standing and the TG and the LTST, the level. When you check the gut, you can find the deletion SK1 can decrease gut permeability. Dextron, we can reflect the gut permeability here, and also can prevent the goblet cell. Is a goblet cell standing, can prevent the goblet cell lost. Particularly, deletion SK1, they can prevent the Th17 accumulation in the gut, in the small intestine lamella propria, and in the large intestine propria. So all of data suggest that S1P induced Th17 maybe contribute to the ALD. Because there are two enzymes, one is SK1, one is SK2. So we wonder maybe SK2 have a similar role in the ALD development. We also try the SK2 knockout mice. However, the deletion SK2 is increase promote the ALD. That's very strange. Is and analyze the reason, the factor. We find the deletion SK2 will cause SK1 high expression. That's the compensation, the expression. So cause the S1P more S1P. So we also use 5-SA to treat the mice because 5-SA is a drug for the treat the IBD. When you use the 5-SA treat the ALG model, you can prevent alcohol-induced liver steatosis here, and also can prevent the SK2 the development of ALD. So this data suggests that intestine inflammation really contributed to the alcohol-induced liver disease. Also, it can prevent the Th17 accumulation in the gut. So this is some work about the S1P in the alcohol liver disease, about the SK1, SK2. Since the fatty acid poly-CoA can drive where sphingosine and go to this pathway can cause the ceramide activation. Our research also focuses on the ceramide. Focus on these ceramides because these ceramides can degrade ceramide to the sphingosine. There are 3 ceramides, like the alkaline ceramides, acid ceramides, and the neutral ceramides. This one, neutral ceramides, is high expression in the gut. In the 2020, there are 2 papers. One is targeting alkaline ceramides can prevent the NASH. And another one is published in Science Translation Medicine. The targeted acid ceramides can increase the YAP1 signal to reduce the fibrosis in the mice. Almost at the same time, we published our paper in November. We targeted the neutral ceramides also can regulate the NASH development, but our work is focusing on the gut environment. We find that maybe ceramides can regulate gut BCL production. So first, we check the neutral ceramides activity in the gut, and in the steatosis in the NASH, we can find that the activity is increased in the NASH. Particularly in the ileum, compared to the liver, neutral ceramides is high expression in the ileum, and further increase in the NASH condition. So we use the neutral ceramides to study knockout mice to study the NASH. We feed the mice with 7 months high-fat diet for induced steatosis and 14 months to induce a NASH, because the high-fat diet very difficult to induce a NASH. So we do 1 year. That's a model. We surprised we didn't find the steatosis affected by the neutral ceramides. From the renal staining and the chest staining, we didn't find any the neutral ceramides can affect the steatosis. However, you can see the liver inflammation is reduced in the knockout mice compared to white mice. In 7 months, you can see many inflammation in mucosal infiltration in the liver environment. Through the muscle staining and the serous red staining, we can clearly show the deletion neutral ceramides can reduce the liver fibrosis. We also check the several the fibrosis gene, like the fibronectin TIM1 or SMA. This gene, all those genes suggest deletion neutral ceramides can reduce the liver fibrosis. Because neutral ceramides is a very important enzyme to degradation ceramide, we think maybe they can affect the sphingolipid metabolites. We do the profile, but we didn't find any significant difference between maybe ceramide. There are many pathways control ceramide production, so maybe we didn't find any difference here. However, we do the lipid profile analyze, we find deletion neutral ceramide can affect monounsaturated fatty acid, particularly in the 16-21, 18-21 in the TG, in the CG, in the phospholipid. You can see the 6-21 TG, CG is decreasing knockout mice, 18-21 TG, CG also phosphorylation decrease in the knockout mice. This data suggests the neutral ceramide may contribute to the MUFA as a production. SCD1 is a very important enzyme to switch such a fatty acid to the MUFA, and the MUFA can promote the TG synthesis and cause the lipid accumulation in the gut, in the liver. So we wonder maybe neutral ceramides can control regulate SCD1 expression in the gut, in the liver. So we check the SCD1 expression and find in the steatosis condition and in the NASH condition, SCD1 already very the decreased in the lockout mice. You can see the significant decrease. And very important in the intestine, SCD1 is almost lost in the lockout mice. And particularly in the MRI level, about 800 difference between the white and lockout. This data suggests that SCD1 is an important factor regulated by the neutral ceramides. So the question is how the neutral ceramides can regulate the expression as CD1 in the gut. One paper published in the Cell Metabolism several years ago, they find that increased the oxalate and contribute to hepatic lipid and liver. SCD1 is both the microbiota dependent. So we think maybe the neutral ceramides, because the neutral ceramides is a high expression in the gut, maybe affect gut microbiota. So we do the gut microbiota analyze. We analyze the live mice. Clearly see the deletion, the mice in the 3 months, the bacterial composition is separated from the white mice. Under the 40 months, you still see the bacterial cluster is separate from the far away from the white mice. Further find the bacteria family, the difference is Ruminococcus family that contains Ruminococcus and Clostridium, and also this bacteria, 3 bacteria genus is high expression in the lockout mice. And very interesting is we find another pathogenic bacteria is a decrease in our lockout mice. We call it the desviral. And in the science paper, they find the desviral increase and decrease cholesterol is a key feature associated with obesity. Our data very consistent with their finding. So we think delete the neutral ceramides maybe regulate the gut microbiota composition and then contribute to SCD1 expression. So in the intestine, gut microbiota is regulated by the IgC, like the IgC assay is a consecration antibacterial peptide can regulate the gut microbiota composition. And also immune system also can regulate gut microbiota, like a BCR secretion IgA can neutralize the gut microbiota. And on the other hand, microbiota also can promote immune system development in the gut, like the PRC IgA development need gut microbiota. So we use the site of analyze all of the immune population in the gut. You can see here very many cluster, CD4, CD8, TCR, neutrophil, and the gamma delta TCR in the gut. We separate the immune cell several cluster, and use and from the site of analyze, we can find the conventional TCR is a decrease. Here the conventional TCR is in expression CD8 alpha, CD8 beta in the gut. And the unconventional TCR is CD8 alpha. This is like the inlaid TCR is very specialized TCR, not like the blood TCR. So deletion neutral ceramides can decrease the conventional TCR in the small intestine and in the large intestine, and also can prevent the Th1 TCR the development. You can see the intergamma on the CD4 TCR is a decrease in the gut. But very interesting, we find the BCR composition also changed. They can suppress the TCR activation, but increase IgA plasma increase in the gut. Like the B220 IgA is the plasma cell. You can see the large, small intestine and large intestine, but this gene do not affect the IgA in the Pierce patch, because Pierce patch is IgA development. There has the germ center area can contribute to the IgA development, so do not affect the PCR in the Pierce patch. Because IgA is the secretion to the lumen can neutralize bacteria. We check the secretory IgA in the lumen, gut lumen. Also, IgA binding the exomolytic particle. All those data suggest the deletion neutral centimeters can increase the IgA binding the exomal or secretory IgA in the lumen. Very important, we also find the IgA binding bacteria, because IgA binding bacteria in the colitis, they can cause the colitis from the patient. You can isolate the IgA binding bacteria from the colitis patient. They can promote the colitis. So we find the deletion neutral centimeter can induce a high amount of IgA binding bacteria in the knockout mice. So we wonder, maybe IgA binding bacteria can promote the SCD1 expression or regulate the MUFA. So to demo this, we use a probe. This probe can detect the bacteria, and this antibody can detect the IgA. So the double staining is suggest IgA binding the bacteria. So we use a fax sorting this cell, and then sorting this cell from the white type mice and the knockout mice, and then transfer to the germ-free mice and give the high-fat diet for the 3 days, 30 days. So you can clearly find the neutral centimeters can regulate the SCD1 expression. When the germ-free mice receive the IgA binding bacteria from the white type mice, can induce SCD1 expression, but not from the IgA binding bacteria from the knockout mice. So IgA binding bacteria is very important to contribute to the SCD1 expression. It's the same. We also analyze the MUFA profile and find the MUFA after germ-free mice receive the IgA binding bacteria can induce the MUFA, but not IgA binding bacteria from the knockout mice. So this data suggests IgA binding bacteria can induce SCD1 expression and promote the MUFA citric fatty acid switch to the MUFA. Because IgA binding bacteria, there are many bacteria. We wonder which bacteria respond to SCD1 expression. So from the bacteria composition analyzed, we find that Rhinococcus is increased in the knockout mice, and the Desfebro, this bacteria decrease. This bacteria is a pathogenic bacteria associated with obesity development. So we transfer these 2 bacteria into the germ-free mice, and the combination with or both to the germ-free mice. We feed the chew dye, a lot of the high-fat dye. You clearly see when you transfer this pathogenic bacteria, you can hugely induce SCD1 expression. But when you combination with Rhinococcus, they can prevent SCD1 expression. That means this beneficial bacteria is a pathogenic bacteria. That's the data suggests nucleus neutral centimeters can regulate the gut bacteria composition, particularly the balance between the beneficial commensal bacteria and the pathogenic bacteria. So next, we also want to know why the MUFA contribute to neurofibrosis. Further, we find the deletion neutral centimeters can affect the wind signal. particularly the Wnt ligand, like a Wnt 3A, 7A, and beta-catenin, and PGSK. All those signal is a beta-catenin signal. So beta-catenin is a very important molecule to promote the fibrosis and liver inflammation. And next, we do some experiment. Use Windows 3A is a ligand that can activate beta-catenin. When you treat the mice, treat the hepatocytes with Windows 3A combination with PUA is MUFA, they can induce the activation beta-catenin signal in the white mice, but not the knockout mice. So all of data suggest MUFA can affect Wnt beta-catenin signal by promote the Windows 3A, enhance Windows 3A this signal. Because Windows 3A is a ligand with a beta-catenin, they can promote this signal, so cause the liver fibrosis. Here is a summary. Is we editorial we publish in Hepatology. The editor, there's comments here. This story is when deletion neutral ceramides can affect the gut environment, particularly affect the B cell produce a secondary IgA. This IgA, secondary IgA, released to the lumen, combining to the gut bacteria, can change the bacterial composition, particularly desirable, this pathogenic bacteria, and the Ruminococcus beneficial commensal bacteria, changes this bacterial composition. And the IgA binding the bacteria can translocation to the liver, cause SCD1 expression, and then switch to the MUFA. This MUFA can affect the beta-catenin signal, promote the inflammation and the fibrosis. So this is the summary. Is we find the gut-derived extracellular vascular can direct the gut and the liver crosstalk. This medium, most medium by the lipid, or like the sphingolipid. So sphingolipid S1P can induce TH17, contribute to the ALD development. And the neutral ceramides can regulate the SCD1 expression and the MUFA the profile and promote the NASH development. Particularly, neutral ceramides in the gut can regulate the gut homeostasis, particularly in the immune cell, in the like the B cell, IgA B cell secretion, and against the microbiota. So it's I like think all of the member in my lab is the past. Red one is past member. Is black one is the current member. We also like I think many collaborators in our university, and like I think the AIDDK support us to study the obesity, and the finding from the AIAAA studies the alcohol-related liver disease. We also get the finding from the AID to study the gut bacterial infection disease. I like stop here. Any question?
Different interventions that might help. So you talked about high-fat diet, but if you is there something in the diet that we can change this? Really change the microbiota you have any specific changes that you see in this pathways?
Yes, it's like we did like the plant drive some the dye, a high fiber dye, and can regulate the contribute to the gut microbiota. We do some I didn't show the work. My previous mentor do the like the plant drive the exome can regulate the gut microbiota, particularly the high fiber dye. But I don't know, we do some special bacteria like the BT that can degradation fiber and produce a short fatty acid, also can contribute to the liver disease. Yeah. Which one?
The flow of the share mode. You can see any questions online.
Oh, share mode. Okay. Let me show and slide. And the slide show. My one question.
So does weight itself contribute to this problem?
Which one?
Now, if you lean versus obese, you look at it.
You mean between the lean and the obesity? Yeah, we do the particle, the exomer like particle, some work. A lot of I didn't talk this project today. Actually, I think we are the first group isolation extravascular like exomer from the fat tissue, and induce the insulin tolerance. We do some work isolating the particle from the fat tissue, and then treat the macrophage and can induce the macrophage activation, like from the fatty acid binding protein. So the exomer, like the particle, is a very important media between the fatty acid, the fatty tissue can secretion many small particle communication with the macrophage in the fatty tissue and promote the insulin tolerance. Yeah, we do some work, and also we do some the plant drive the particle in the obesity and affect the gut microbiota. Yeah. Okay.
So is it like both ways in the sense that the gut microbiome is affecting obesity, but obesity is also affecting the gut microbiome?
That's is maybe interaction. We maybe don't know which one is. Some bacteria is affected by obesity, but some bacteria is promote the obesity. They can, like the Destrone viral. We talk about this pathogenic bacteria. I think there is a causative factor can promote obesity, particularly insulin intolerance. They also do the fatty liver disease study, the bacteria, because some bacteria can secretion tolerance receptor ligand, and then activation the Kupffer cell in the liver. So all secretion the like gut bacteria can secrete the ligand is the tolerance receptor file, a flagellum. They can also promote the fatty liver disease. So yeah, like they can switch the fatty acid composition from the tolerance receptor ligand. Yeah.
Is there any impact of diabetes on this?
Yeah, we also check some model is use check the insulin tolerance, not only obesity. We also check the insulin tolerance with like we talk about the sphingolipid also can affect the insulin tolerance. We didn't show that data because our model is global knockout. Right now we generating the intensity epithelial cell and specifically target this gene in the gut. We try to find a specialized the lipid can contribute to insulin tolerance, like because gut can secretion many ceramide. The ceramide can migration to the liver, can cause the sphingolipid. And also we try to find which type of the sphingolipid, because many sphingolipid can modify by the glycan. So our work also focuses on the glycan-related sphingolipid. So try to find this lipid contribute to insulin tolerance and obesity development. So one of the R1 is last year is talk about the glycan related sphingolipid. Yeah. Okay.
Which one? This one?
Yeah.
This one.
Yeah. This. Yeah.
I think I have.
Okay.
So yeah, I did talk about the diabetes and the insulin tolerance. Actually, my work is also very interesting. So we like to get some collaborator with because my work also AID can benefit to you and me. So we want to write a proposal about the diabetes. My first paper about I think is published in the diabetes is talk about the how exomal induce macrophage activation and cause insulin tolerance. So I'm very interested into the how gut drive factor in not only affect the fat liver, but also communication with the fat tissue. There are some very lower ideas neural affect fat tissue. I'm not sure you are familiar. My some work last year we published one paper is enteric neuron. It's a neuron in the gut. I interested in neural gut, how neural gut affect the fat tissue. That's so maybe we can check
because there is one particular group that has been looking at